Today we have Emily and Andrew from the Phi Lab. They're going to be talking about unsupervised discovery of neural sequences and large-scale recordings. And this is just to sort of remind you all, I guess, this is a computational tutorial. So they're organized by grad students in the department. We're always looking for, I guess, new topics and people to give talks. And so if you personally have developed some sort of a method that you think would be really good for presenting in this type of a setting, or if you are interested in inviting somebody to talk about a method that you really want to start using, talk to me, send me an email. You can find my email on the event announcements. And then I guess the last thing is that there are GitHub slides, or there's a GitHub and then slides, which are also on the event announcement, which you might want to get later. I'm sure that Emily and Andrew will also give you some directions when you might want to pull those up. Great, so I'll turn it over to them.
Thanks. So the lights, I think we have two options, dark and bright. And
so we're going there might be a few dark slides that are hard to see, but just bear with it, and hopefully most of them will be clear.
What if we turn these lights off?
So either all of them are off.
There's like only presets,
so we can all sit in the dark.
But I think if there's anything you can't see, ask about it, but we tried to check, and hopefully there's enough contrast that you can see it. So thanks, Janelle, and I guess I want to reiterate what Janelle was saying about the tutorial series, is that if anyone has any topics, this series is very sort of informally run. If there's a topic that's of interest to you and you want to run a tutorial or invite somebody to run a tutorial, please do that. So today we're going to be talking about algorithms for detecting sequences in high-dimensional data sets. And so the goal of this tutorial is to give you an idea of whether these methods might work on your data set and some tools to get started on that. So as Janelle mentioned, we posted the code and we'll describe sort of the steps involved in it. And the algorithms that we're describing are direct extensions of some very simple dimensionality reduction techniques. And then there have been some elegant extensions of those to the time domain, which were started in the acoustics fields. And we found that they work really well on neural data, and we've made some of our own extensions. And so that's what we'll be showing you today. Okay.
In addition, we also have a bioarchive preprint, so if there's anything that you see here and you want more detail, there's some extra detail there.
Yes.
Okay, so neurons form sequences in many different brain areas, but I'll start by showing you our favorite example. So here in the rows are different neurons. Each neuron has a different color, and within each color, there are different rows corresponding to different renditions of this syllable shown at the top. And so for an individual neuron, you can see that the firing is very robustly locked to a particular time with respect to the behavior. And if you look at a population of these neurons as a whole in the area HVC and the songbird, you can see that together they form this sparse sequential structure. And although many of the examples that we're going to show you today may look like this structure here, these methods work very well not only on sparse sequences, but also on any sort of repeated spatiotemporal pattern. And in other parts of cortex besides HVC, which might not only generate these sparse sequences, we think that there's lots of examples of repeated spatiotemporal structure. And some of our favorite examples, I'll go through some of our favorite examples here. So one example comes from the hippocampal field. So here again, there are firing rates of neurons in each row. And then on the x-axis is time during some behavioral, the onset of some behavioral event. And then if you look at the firing rate of these neurons throughout the behavior, you see that they form this sparse sequence again. And likewise in PPC, in the rodent, during another task, people have reported these sparse sequential firing, which is locked to different types of behavioral tasks. And then stepping away from just neural firing rates, here is shown the principal components of a neural ensemble, the first three principal components, as well as trajectories through those three principal components from the Jaziri lab here. And so even if it's not really just a simple sparse chain-like organization of neural activity, you can still imagine that here, as the neural ensemble goes through this trajectory, that they visit a repeatable set of states, and that things like this we might want to uncover. And then finally, in recurrent artificial neural networks, after training, many of these networks will exhibit sequences of dynamics given the same input. So there are really a lot of different places that we see sequences in neuroscience. And in addition to the sequences that we see in the brain,
oh, sorry, I just wanted to interrupt very briefly. I just wanted to ask of what type of data sets people in the audience have with sequences that you're looking at, just so we can get an idea of this.
Hippocampal replay sequences.
Okay, hippocampal replay, cool.
Calcium imaging.
Calcium imaging, where?
Hippocampus.
Hippocampus.
Cool.
Okay.
Speech signal.
Speech signals.
Awesome. We'll
Okay.
talk about spectrograms in a bit.
Okay, cool. So, which is a perfect segue. In addition to these neural signals, a lot of us neuroscientists and cognitive scientists, are interested in behaviors. And these behaviors can also have robust, repeated sequential dynamics that you might want to extract or study. So the way that we got interested in this was from some of Emily's data that she'll tell you about now.
Yeah, so I'm also doing calcium imaging. What you're about to see here is fluorescence traces extracted from an endoscope recording in songbird HVC, premotor cortex, the same area that you saw sequences in briefly. So this is in a young bird singing. And so you can see some patterns of activity happening. And so, yeah, this is why I got interested in this problem of extracting sequences, because I wanted to extract sequences from this type of data. And so actually a previous one of these tutorials was given by Pencheng Zhu from Paninsky Lab, and we've been using his algorithm to go from the raw movies to extract the temporal traces of neurons. And so then you get a data matrix which looks like this. So each row is the functional calcium signal from a different neuron over time. And I'm going to briefly show you our algorithm running, just to show you a sense of the goal, and then we'll explain it in more detail. So the goal is to extract repeating neural patterns and also extract the time courses, the time points at which those patterns occur. So I'm going to show you it running, and on every iteration, I'm going to reorder the neurons according to their location within each pattern.
I just have a question about the data. Is this the deconvolved spiking activity, or is this actually still just calcium transients we're looking at?
This here is deconvolved activity. You can try it on either. And it could work on either. It depends on sort of the length of your sequences. Yeah. But this is the deconvolved. Yeah.
But yeah, we've also tried it on the raw calcium, or just the calcium traces, and it works there as well.
So you can see that the neurons are being resorted into patterns, and so it's extracting these repeated patterns of neural firing and also the times at which each pattern occurs. So you can see, for example, this yellow one happens at these times. And this is the same as the same data just resorted, and you can see there's a lot more structure in it now. And if you go back to a spectrogram of the bird's song, you can see that each of these neural sequences happens at a point when the bird is singing a particular syllable. So this is correlated with behavior, but we didn't use the behavior to find these sequences. We found them directly from the neural data. Okay, so how does this work? First, going to give a bit of a review of matrix factorization, so NMF and PCA. So let's say we have this data matrix, neurons by time, and our neurons fire in a synchronous ensemble. We might want to extract a pattern and also the times at which that pattern occurs. And we can use these, convolving them to reconstruct our data. So place this pattern at each of these points. And this is non-negative matrix factorization. It's a similar framework to PCA, but everything is required to be positive. And it generalizes as well. I think, yeah, that just says NMF. It generalizes as well to if your data matrix had another pattern in it. Then you could just add another factor and the times at which that factor occurs. And you can reconstruct your whole data matrix by adding together the reconstruction of pattern 1 plus the reconstruction of pattern 2. And you can really succinctly write this down as the matrix containing all of the patterns, matrix multiplied by the matrix containing all of the time courses. And they're simple algorithms developed by Li and Sung for estimating what the patterns are and the times at which they occur. And so you can do this by starting off with a random set of patterns and time courses, and then iteratively either fixing the time courses, these H's, and updating your W's, and then fixing your W's and updating your H's. And this is really elegant and simple algorithm. It's easy to code up. Andrew and I have both done it, trying to get intuition about it. It does not take very long. So I'd suggest that in the exercises if you're interested in that. Okay, so this is the non-negative matrix factorization framework. The problem with it for the types of data that we're interested in is it doesn't work on sequences. Question?
Yeah.
What were the H and X matrices?
Yeah.
So what was the X?
So X is the data matrix.
Yeah, this is X.
Yeah.
So if you go back to the updates, this is X, and then WH is your estimate of X, which we'll refer to as X tilde. And so you are using W and how it overlaps with your X and your X tilde to estimate H. And this is just gradient descent, but you've set a learning rate so that everything is always positive.
It's essentially a trick to make sure that you don't have to do anything fancy to keep things positive, was to rearrange things so that you only are doing multiplications.
Yeah, and it's a very simple derivation as well. It's I think it's in Li and Sung, and then we also reproduce it in the appendix of our bioarchive thing. But it's okay, so let's say that you tried to apply NMF to this type of data. What would happen? No ideas?
Mm-hmm. Is it going to fail because of all the singularities, like all the blanks? It's going to make it non-singular.
Yeah, I think what you might be getting at is that like at any given point, only one neuron is firing, so there's no synchronous patterns that you're able to pull out. And so the best you can do with any factor here is just pulling out one single neuron. You can't pull out any ensembles of neurons firing together because literally in this type of data, the neurons do not have any overlap in time of their firing. So what we really want instead is to pull out a pattern that's allowed to last some short amount of time. We're going to call this time length L. And then if we have this, we can do a similar thing with NMF. We can just convolve that pattern with a time trace, which effectively places this pattern at every point where it happens, and we can reconstruct the data that way. And this is a technique called convolutional non-negative matrix factorization, which was developed by Paris Smaragdis. And it, like NMF, you can generalize it to multiple patterns. So you can just, if you have multiple sequences in your data, you just add up the contribution from factor 1 plus the contribution from factor 2 and use as many as you need. And like NMF, it can be fit with simple gradient descent. So the cost function is you're trying to make your reconstruction of the data, which is like matrix multiplication in the NMF case, but you have a convolution in here now because you're convolving each of your patterns with a time trace, and you want to make this reconstruction match your data. So this can be done also with the multiplicative update rules. So do you have the H's? So this is similar to what you saw for NMF. It's just done separately for each of these L points. And so, and similarly for the W, you update each of the time lags separately. And so this works very well for extracting these kinds of patterns. The problem that we ran into when we were first trying to apply this is that especially when you don't know how many sequences exist in your data, you tend to get redundant factorizations. So I'll show an example of what I mean by this is here's the data, but it's now factorized into two sequences, the first half of the sequence and the second half of the sequence. And this can be hard to interpret because each time you run it, you'll get a slightly different set of sequences. And it's sort of an inefficient way of representing your data because you could have used one sequence factor to describe it, but instead you're using two. So we wanted to prevent this type of thing from happening. And we'll show you a couple ways of preventing it. So I'll start with the first one, which is basically noticing that in this case, you have a lot of correlations between your different factors. So these two, the H's are correlated with each other. So we want to add a term to the cost function, which penalizes correlations. So the first thing to try would be to add the correlation matrix, HH transpose, to the cost function. And this has been done on NMF and works fairly well. The only problem here is that HH transpose only catches correlations that happen at instantaneous time lag. It doesn't capture cross correlations that happen at some delay like these happen. So you miss that. And so the first thing we tried to get around that is adding a smoothing matrix. So if a smoothed version of H is correlated with H, that would capture this type of correlation. And that works pretty well, and it actually gets rid of this type of redundancy. But the problem there is that now you get this type of redundancy. So here, each instance of the sequence is explained by a different factor, but they are redundant because you could have just used one each time. And so in order to get around this issue, we noticed that basically these factors are themselves overlapping with the data at similar points in time. So what we're doing is what we're going to do is measure the overlap of each factor with the data and compete those against each other. So say that the overlap of factor 1 with the data should not be correlated with factor 2. So this here is the overlap of the W with the data. And we're looking at the correlation matrix between that and the H's.
How is that different from just correlating W1 and W2?
That's a really good question. How's it different from correlating W1 and W2? It's related, but it also gets at correlations in H's. So you could have a case, like the first case that I showed you, the W's are not correlated, but the H's are. And so this term sort of symmetric in W and H, and so it gets at both H correlations and W correlations. And we'll come back to the options of just punishing H correlations or just punishing W correlations in a bit.
The other reason that you might want to do that is that here, the size of W, L, is just wide enough to fit that sequence, one instance of the sequence. But if you imagine that we extended L to be slightly larger, then you could shift those sequences. So they could be offset from one another, and then you could shift the H's. So you could have two factors that are reconstructing the same data, but if you were to just look at the patterns in the W, they'll be offset from each other.
Yeah, basically it's the process of developing it. It seems like we think we come up with a clever idea for fixing it, and then it dodges it in like a cleverer way. And so, yeah, there's a lot of ways that it could create a redundancy. Mm-hmm.
I just have so in this idealized case, let's say that's your actual data set. These issues wouldn't come up, right? Is it like, so is this due to the noisiness of neural data, or is it like
this actually happens in noiseless data?
Okay.
And the reason is that if you don't have this in your cost function, if you're just trying to reconstruct your data, then each of these redundant solutions is a good solution. And so it's like your cost function has all of these different but equivalently good global minima. And so it just randomly picks one based on the initial conditions, and what we're doing with the penalty term is sort of tipping it to favor the simplest of those. Okay, so this also, adding regularization can be easily incorporated into these multiplicative update rules. Li and Sung noticed this when they first developed the regularization terms. So if you remember, which you might not unless you have a really good memory, this is just the same as the convolutional one, except we've added this extra term, which is the derivative of the regulars or the penalty term. So this is also fairly simple to implement in MATLAB. Mm-hmm. Okay.
Okay, so the first thing that we wanted to do was to systematically test this algorithm on a couple of different types of synthetic data. So this is just some very simple model data with two sequences and a little bit of noise added. And when you run this algorithm with the penalty, then you recover the two patterns in the data as well as those times at which they occur. And you can change this data slightly to add neurons that are shared between the two sequences. So here on the top, you can see that each sequence pattern has a subset of neurons that are unique to it, as well as a set that are shared. And it can...
They're the unique ones, and these are the shared ones.
Mm-hmm. And it can extract those patterns just fine. And then alternatively, on the bottom, you can see a very similar example, but here the neurons that are shared across the sequences have different latencies that they're active in each pattern. And so this method can work very well at these sort of simple cases, and it doesn't matter if you have neurons that are shared between them. And one way that you can quantify this is by looking at consistency. And so consistency here is the measure to which two different factorizations, in two different factorizations, there's a good one-to-one match sort of between each of the factors. And so, and then you can see probability of observing a particular consistency on the y-axis. So here in orange are all of the unpenalized factorizations, and then in blue are the seqNMF factorizations. So you can see that there's a large improvement in the consistency in that you tend to get very similar factorizations on the same data if you run it many times.
What data set is this from?
So this is from a similar data set to one that's shown here.
Generated data set.
Yeah, this one is shown just for a model data set. And one thing that you might want to do is after you've, what's that?
Oh, I thought there was a question.
Okay.
Oh, you
Yeah?
do.
So how do we set the lambda parameter there? Are you using cross-validation to set the lambda?
Good question. We'll get into that.
Yeah. So one thing that you might want to do after you've fit your data and you've got some factorization is you might want to just test if it generalizes to another data set. And so here you could imagine that you have some data that you held out, and you have your factorization that you obtained from fitting the first half of your data. And now you want to just see if it's still present in that second half. And so you could imagine sliding your pattern across the data and looking at its correlation with the data at many different times. And then you could make a null factor by taking each of the rows and shifting by some random amount. And you could do that many times and look at the correlation with the data, and you'll get some other distribution of correlations. And so we used a method like this to test which factors in our factorization were significant. And so here you see on the y-axis the number of significant factors in a given factorization with 20 factors. And then on the x-axis you see that we repeated this for model data that has between 1 and 10 different sequences. And then you can see that when you use this penalty that you obtain a number of significant factors as assessed on this held out data that's very close to the true number that we put in the synthetic data.
And if you don't have the penalty, then because there's a lot of redundant factors, there's a lot of, like basically almost all of the factors are significant on held out data because they're just redundant copies.
Okay, so we also wanted to test this algorithm on model data that had a few different types of noise that we expected to see in neural data. And so here I'm going to show on the top just a snippet of the data that a factorization was fit on, and then below is an example factor that was extracted. So here is data that has 3 sequences in it, but neurons only participate in a given sequence with some probability. So you can see that you can still extract a sequence from data that looks like this. You can also add noise on top of it, just add additive noise, and see that it can extract things just fine in that type of noise. You could also imagine jittering every neuron in a given sequence by some amount. And so here you can see that the algorithm can still extract patterns even in noise like this. And then finally, we tested in data where the speed of a given sequence is fixed within the sequence, but it randomly speeds up or slows down. So sometimes the sequence occurs at one speed, but sometimes it occurs up to 5 times slower, for instance.
And one thing to say here is that it almost looks like it is sort of denoising it, so it's only extracting the part of the data that's repeatable and ignoring all of the noise. And so we're going to get back to that idea in the end about how you might be able to use this type of technique for denoising. Mm-hmm.
Do you specify from the start how many sequences you expect to find? Is that apparent?
No, so that we're going to get into how to estimate that, basically. Yeah. Mm-hmm.
What would denoising mean in the context of the warping example? Because
that's a really good question. Yeah, so it depends what you want. So you might, in the case of warping, what would denoising mean? You might think that warping is just noise that you don't care about, so you want to go to an average unwarped case, but you might care about the warping noise, in which case it's not noise, it's signal. And effectively, you can sort of change the strength of the penalty to go between these cases. So if you have a very strong penalty, or reasonably strong penalty, you'll get 3 sequences back in the warping case. But if you weaken it a little bit, then you'll get sequences for slower and faster versions of the sequence. So it depends sort of what granularity you're trying to describe your data at.
I might not fully understand the algorithm, but it seems like it almost, in the extreme case of warping it, it can't work because you have something that's purely synchronous versus like flat. Like you would need multiple factors.
Yeah, the warping is sort of like an interesting fringe case. It's not really designed for warping. Like if you really want to study warping, you should basically have a model of the data that incorporates the speed into it.
Or you could change the time basis for behavioral states, like I'm thinking sleep versus.
So you could warp to a behavioral variable like position or something like that. This is just saying if you don't care about, if to you warping is noise, then it will ignore it. But if warping is signal, you should probably use something else. Yeah.
Yeah.
Mm-hmm.
What does it?
Yep.
What does it look like when you run it on, say, like pure noise? Like, what would
Pure noise. I wonder if we have an example.
That's a good question. Yeah, it will often pull out a factor that either explains one single neuron or one single instant in time, and these factors won't be significant on held out data.
Depends a bit how long your data is. If your data is very long, then it will usually choose a single neuron, whatever the highest variance neuron is. But if your data is fairly short, it will choose the highest variance time, and it will sort of trade off between these things. Yeah.
You mean cross-validation would rule it out to like discover accidentally like the data?
Yeah.
Basically.
Yeah, so if you had a separate, like let's say that it pulled out one instance of time, which is sort of a frozen instance of noise, and then, because that explains some variance in your test set, if you looked at how well that overlaps with held out data, it's not going to be significantly different from the shuffled version. Mm-hmm.
So if this was like neuron data, this is the rate function, or is the actual spike times?
Good question. We've you'd probably do rate. You could do both. Yeah. I guess one thing in terms of like what data should you plug into it, you don't need to bin it as finely as spike times. Like if you think that the neural events are sort of broad, then you could plug in sort of smooth spike times, for example.
Yeah, it depends. We haven't extensively tried it with spike times, but you could certainly do that. But for instance, the 2 data sets that we spent the most time working with, which is a hippocampal data set and the Emily Songbird data set, the time at which the sequence unfold vary by like an order of like they vary by one unfolds at the order of 20 seconds and one at 100 milliseconds. So you just have to be careful about how exactly you process your data. Yeah. Okay, so what we wanted to do with each of these, so clearly in all of the extremes of these noise cases, this algorithm can't work at all. So we just simply tested how far we could go out in each noise case. And the way that we assessed this is that first we created a noiseless data set, which is to say a data set that looks like sort of the first data set at the top left here, but with all the neurons participating. And then a bunch of different data sets with different amounts of noise. So here you can see as we go to the right, increasing number of neurons are dropping out on every instance of the sequence. And then to assess how well the algorithm was recovering the original data, we looked at the correlation between the sequences that were recovered by the factorization and then the original sequences that were used to generate the data before adding any noise. And so here you can see for many runs, the similarity to the ground truth sequences as a function of the percent of the neurons that are dropping out on any given instance of the sequence. So of course, when it's very low, you usually get a very, very good match, close to 1. And then as you go out to about 60 or 70% dropout, you start to get a drop off. And then of course, by the time you're over to the data that set at the very far right, it's very difficult. There's essentially no sequences there, so you really don't detect anything.
Yeah, so it's quite robust in the sense like it's failing, but it's failing when you would look at that and it doesn't really even look like there are sequences there.
So this is just for the case of the participatory.
Yeah, so I'll show you now all the different types. So this is the same analysis for each of the different noise types. So at the top, again, you can see 3 different example data sets. And then at the bottom, for that same level of noise, is one of the extracted factors for those 3. So you can see at the bottom that, for instance, in the additive noise case, the second one, you can see that even when you get very deep into the noise, you can still get sequences that are qualitatively very similar to the ground truth sequence, even though perhaps the amplitude of it might be very suppressed. But for something like very high jitter, like you can see at the in the third column, the top right data set, there it's unclear whether or not we should call that a sequence, even though it was generated by forming a sequence and then jittering things. And there you can't really recover anything. And again, for the warping case, it depends on the parameters that you're using when you're fitting, and you might be able to still recover 3 sequences that look somewhat like the ground truth, or you might end up with many, many different sequences at different speeds.
Yeah, and the warping, it'll sort of take the average of them.
Yep.
How are X and Y being treated in this case?
X.
Is it just, I mean, is that a parameter, I guess, that you can specify that you want it to be convolutional in both time and frequency? Or I guess what's happening?
We've
with these
only
examples?
done convolutional in time.
Okay.
Yeah, or in
so
one dimension.
In all of these, it's like neurons by time.
But you could imagine it being anything by anything, but we've only worked on the one-dimensional case where you're convolving only along the one.
Sorry, so then what are the sequences that it's picking out here in relation to
That's
the
a good
top?
point. This is just one example sequence. It pulls out 3 sequences, one that would be
Yeah.
these neurons,
Exa-
one that would be
exactly.
these neurons, and one that would be these.
Okay,
Yeah,
it
that's
just
a good
happens- it
question.
just happens
This
like-
is just one example to give a sense
I just
of it.
plotted
Yeah,
the same one so you can compare, yes. But it will extract 3 different sequences, and the similarity measure looks at all 3 of those to see how well you did on the complete set.
So for the jitter case again, you're just jittering each element in the sequence with
Yeah.
the same range.
Yeah, exactly. So for each time a sequence occurs, there's some distribution of jitter times, some Gaussian distribution of jitter times, and you just draw from it and jitter by that. Exactly. Yeah, so that's what's shown at the bottom is the standard deviation of that distribution. Yeah.
So are you worried about the robustness in the dropout case? Because here you're in control. You're the one generating these sequences, but in the brain you don't. Those sequences that are missing certain neurons could actually have different meanings. Like it could be.
Could.
And maybe the regularization you're doing is kind of really biased against that.
Yeah, no, that's a good question. And what we've started doing is sort of looking at different, like when you have a sequence, what does each instance of it look like? Does each instance actually look like the average, or does it look like something different? Like, is there a lot of dropout? And you can also look at, for each neuron, does it always participate in every instance? So these are sort of the analyses that you could do once you have your sequence.
Yeah, and we'll talk a little bit about that because that's exactly something that we're interested in. But I guess for the dropout, I mean, these data sets, in order for this to work, have to be pretty long. So I'm just showing a very small snippet of a much longer data set. So if it's really random dropout, like in this case, then you might expect it to work. But if there's something systematic about the way that it drops out and you have enough data, then you should be able to tell the difference. Okay, so we're fairly convinced that it can work in a few different types of noise, at least at a moderate level, and give us sensible factorizations. But we also wanted to just see sort of how much data do we need to put in to get a qualitatively good factorization. So here is a short data set. This is just 400 bins of the synthetic data, and it just has one example of each of the 3 sequences embedded in some noise. And so first, we're going to just factorize this very small data set, and then we're going to iteratively increase it by another 400 bins and keep going out and see how well we can factorize. And so here on the x-axis is this increasing number of bins in a data set, and then on y is the similarity of the extracted factors to the ground truth in this noisy case. So this corresponds to one of the violins in the violin plot that I previously showed you. And then at the bottom again, you can see factors, W's, just one example of the factors that were extracted at the bottom. So you can see that for a data set that has 400 bins in it, you don't get a very good similarity, but you can already see the sequence a little bit in the factor that was extracted. But by the time you go up a 3 or 6-fold increase in the data size, then you start to do pretty well.
And here you just have 3 examples of each sequence, and it looks a bit noisy, but you can definitely see it.
Yeah. Okay, so one thing that is an important question is how do you choose your parameters for this model? And so there's many different ways that you could think about how to choose the parameter, but it's very important to figure out exactly what value of lambda you're going to use, and we'll just walk through sort of a couple of approaches today about how we've done this, but here's one of them. So when you fit the data, here we fit the data with lambda spanning a few different orders of magnitude, and in red is shown just the reconstruction error on the data, and normalized to go between 0 and 1. And the blue is showing the correlation cost, also normalized to go between 0 and 1, where the correlation cost is the value of our penalty term at each of these lambdas at the end of the fitting. And so here you can see that if lambda is really low, then your reconstruction error saturates at the sort of best reconstruction that you could ever do. And here in noisy data, you'll be fitting a lot of the noise.
So if your lambda is low, then it's this reconstruction term in your cost function that's being prioritized. And then on the flip side, when lambda is really high, it's just the correlation that's being prioritized, and you're getting a lot of reconstruction error. And in the extreme case, when lambda is really high, everything has just been collapsed onto one factor. So there's no correlation because there's only one factor.
Yeah, so you have some number of factors in your model, and on the right, you're only using one, and on the left, you're using all of them as much as possible to fit the noise. Okay, so what we wanted to do is see if we could use these measures to figure out a good way to choose lambda. And so here I'm showing the same thing for one of the intermediate level noise cases that I showed you before on synthetic data. And so again, you can see that it saturates at high lambda for the reconstruction cost, and it saturates for correlation cost at low lambdas. And then in the...
So this was a 3-sequence case, and this is showing how many significant factors there were. And you can see there's a sort of plateau at 3 for a range of lambdas that's spanning a couple orders of magnitude. But if you go too high or too low, you're not getting the correct number. And this is the percent correct of those. So that's that region where that was 3. And then if you look at the similarity of these factors to the ground truth factors, likewise, you see a plateau. And this is just showing the product of those two curves to get an estimate of this good region of lambdas. And so then what Andrew did was compare this region to that plot for all of the different noise conditions and see if we can systematically say relative to this plot, relative to that crossover point between the correlation cost and the reconstruction cost, is there a systematic way of picking lambda?
Mm-hmm. And so we're calling this crossover point. Here you can see lambda not. And so here is sort of a summary of the ratio of the best lambda as assessed with the percent correct fits and the similarity. So we're calling the peak of that where you have the right number of significant factors and also a very good or the best possible match to your ground truth factors. We're calling that sort of the best lambda. And then we're asking how it's related to lambda not that crossover point. And so each color here shows a different noise condition. And as you go from left to right, you're increasing the amount of noise. And so first we have in blue is the participation, and then in red is the additive noise, green is the jitter, and purple shows the warping. And so you can see that for each of these conditions, there tends to be between sort of 1 or 2 order of magnitude range that you can use and still get a very good match to your data. And here the colored lines, the bottom and the top of the color lines show the half maximum crossing for this similarity correct fit score, this plot.
So the basic intuition is if you try lambdas somewhere at around 2 or 5 lambda naught, then you should be able to see the right answer.
Okay, so next we wanted to look at a data set where we were already pretty sure that there were neural sequences. And so the analysis that we're going to show you now comes from a data set from Yuri Buzaki's lab at NYU, or at the time I guess it was at Rutgers. And so in this data set, there's a rat that's running on this figure-8 maze for a reward. And so it has to run alternating between left turns and right turns. And in the period between the left and right turns, it needs to run on a wheel for about a 20-second delay period. And so in 2008, in the Poslikova et al. Paper, they observed that during this delay period, there's sequential firing of hippocampal neurons specifically during this running wheel period.
And some, go ahead. No, in some of their rats, they saw just one sequence in the running wheel, and in some of them, they saw different sequences, whether the animal was about to take a leftward trajectory or a rightward trajectory. So this was our test case to see if we could pull this out.
And the great thing is that all this data is on CRCNS, the neuroscience data server that Yuri is very involved in, so it was super easy to just download it and try it out. So this is sort of the first rat that we tried. So there are several rats online, and on the right you can see the raw data sorted by the factorization. And then on the left you can see the reconstruction of the data as well as the factor that it pulled out with the H. And you can see even in the raw data after sorting it that there are these 3 sequences, at least in this particular data snippet. And if we look at the behavior of the animal, here shown in red, is a plot that just shows you when the delay period is, and you can see that the H and the sequence that were extracted are very robustly locked to this delay period.
So the sequence that was extracted happened during the delay period, and then a little bit right after when he's running around, running through the track.
Okay, so we can also try this for another rat, and in this other rat we were able to observe multiple sequences. So here you can see the same thing. This is the first of 3 sequences that were extracted. On the left is the sequence again, and then the reconstruction of just that sequence, as well as the sorted data by that sequence is firing. And then for a second factor, you can see that it alternates between this purple and red factor, just like the behavior where there's an alternation between left and right turns. And then if you look at a third factor, you can see that this yellow-orange factor comes after both the purple and the red factor. And so this corresponds to the rat running down the stem of the maze right after the delay period ends. And so these are probably place cells, but the animal's behavior is so stereotyped that you can actually extract a sequence during this time as well. And so here I'm just showing the way that we chose lambda here, as well as the significant factors. So using the same procedure that I was just showing, we found lambda naught, and then we used our calibration with the synthetic data to see how many how much larger we should make lambda approximately. And then on the right is shown just the histogram of the number of significant factors from many runs on the same data. So you can see for the first rat, most of the time you extract only one significant factor, although sometimes two. And so this probably corresponds to sometimes the delay period sequence and the sequences the animal runs on the stem of the maze are put together, and then occasionally they're split apart. And then for the second rat, you can see that usually we're extracting 2 or 3 and sometimes 4. And this is corresponding to the left, the right turns, and also the stem of the maze.
Mm-hmm.
Looks like the time scale of the rat data set and the bird data set is quite different. So I was wondering if you have to change the width of W significantly for this. Or how
Yeah,
much
you
is-
need to change the L, basically. So this is a
Right, the delay.
this is L. It's a number of time bins to use in the factor, and basically the strategy for that is just choose it slightly bigger than the biggest sequence you're expecting in your data set.
What if you don't have any a priori kind of knowledge about your sequences? What would be the strategy? Like you have a nice strategy for lambda, but do you have a sort of a
Yeah, if you basically need to know something about the length of the sequences that you're extracting. But I guess the caveat to that is if you just make it big and there aren't any long sequences, there are only short sequences, what it'll do is it'll leave all the time bins empty except for the ones in the middle for the short sequence. So that's a good strategy to try. Like, it's okay to make it slightly longer than you want. The algorithm might take a little bit longer to run because it's running over more L's, but it'll just leave the outside empty.
Yeah,
For
but
a lot of us, like the question way before that is like whether or not there is any sequence in the data set. We kind of, if like there is there any like parameters like byproduct during this like fitting, you can like likelihood or whatever to tell like how sure you are this can explain the data or something. That would be like nice.
Yeah, so I guess there's a couple questions bundled into that about like how do you choose the length and also how do you test the significance. And so for the length, I would just choose something long, and then if you see sequences that are shorter, just shorten it if you want to. And you might have some prior information. For example, if your animal is doing a task that lasts 20 seconds, then you might expect sequences that last 20 seconds. And in terms of cross-validating and testing significance, we described the significance of individual factors, but we're going to go into an interesting method of masking, basically. So there's a way of cross-validating your entire extraction and using this to estimate how many sequences might be in the data. So that's coming in a few slides, yeah.
Yeah, but if you really don't have an idea about how long your sequences are, there's nothing sort of special that this algorithm can. If you don't even know an order of magnitude, then I don't think we have a way to help you using this. You really have to be sort
you
of
could
in the
try a
ballpark.
few different ones, and you could say like, well, there weren't sequences at this short one, there weren't sequences at this long one, but you need to have some guess of the length, roughly. Yeah. Okay, so this is just another songbird data set, and using the same procedure to choose lambda. We often got 3 sequences, and this is what those sequences look like. This is the resorted data with these 3 factors, and the reconstruction of the data with these 3 factors. And you can sort of see that each of the factors correlates with particular song syllables again. Okay. And then we were interested basically because we have a lot of spectrograms, and this algorithm was initially developed for spectrograms. So convolutional NMF was initially developed for spectrograms, and we were wondering whether it could extract structure in the spectrograms of juvenile birds. So this is previously published song recording with hand-labeled syllables because they can be difficult to label in young birds. And when we ran seqNMF on this data set, it extracted factors that closely matched the hand-labeled syllables. And it even extracted an extra syllable that basically in the hand labeling, it hadn't been clear how to label it. So yeah, this is just showing the correspondence between these factors and the hand-labeled syllables and another syllable that was unlabeled by hand, but it pulled it out. Okay, so basically, we think it could work on many of your data sets. And so this is the sort of just giving you, what we've already done is giving you a sense on of the type of data sets that the algorithm might work on. And then in the remaining slides, we're going to show you some extra tools. So tools for cross-validating, tools for choosing between different types of factorizations when the answer might be ambiguous. And then we're going to discuss denoising and link to our code and the exercises.
Okay, so there's a sort of simple cross-validation procedure that works on a lot of different matrix factorization approaches like PCA, SVD, NMF. I'll just walk through it very quickly. So the idea is that you have some data matrix, and you're going to just hold out a random subset of them. So here, shown in white, are the masked elements that we're not going to fit in our model.
And one thing to note here is that for unsupervised techniques, you often can't really have a separate held out set, because to measure whether your separate held out set, whether you've overfit, you have to then fit your factors on the separate held out set. So this is a bit of a nuanced point, but Alex Williams has a really great blog post about this and about doing this type of cross-validation with leaving out random bins in your fit, and how it can be applied to a lot of unsupervised techniques like PCA and NMF.
Yeah, you can imagine that if you don't fit the second half of the data, you have no time course, you have no H, and you need an H in order to assess your fit. And so now you're stuck with the option to either fit your held out data set or to come up with something clever that will work also. And so here you're going to hold out these random speckle elements, and now you're going to perform your minimization, but during the minimization, you're going to continue to mask those elements, so you're not going to minimize the reconstruction error on any of those white elements, only on the gray ones. But now when you're finished, you're going to look at those elements now, and you're going to see, even though I didn't fit them and I didn't perform the minimization on those elements, did I still get good generalization there? Do I still minimize the error there? And okay, so that works really well for k-means, PCA, SVD, NMF, and also you can do the same thing for our convolutional NMF case shown here. It works just the same way. You just mask the elements during the fitting, and then you're just going to look at that subset of elements once you've finished with the fit.
And what we're going to use this to do is to have an estimate for how many sequences there might be, because we want to see for different values of k, the number of factors, when do you start overfitting? So you should, if you add another factor, you should get better and better, and then at some point you're just fitting the noise and you start getting worse on the held out data.
Okay, so here's a synthetic data set, and shown in blue are those gray elements, the ones that you were minimizing the error in. That's our training error. And then in red is shown the RMSE on just those elements that were masked during the process. And so here in this data set at the top, you can see that there are 3 sequences embedded in some noise, and that as we increase the number of components in our model, we get a minima in our generalization error in red at 3. And that as soon as you go above 3 components to 4, you start to overfit massively on your noise. And as a result, when you look at those masked elements, you're going to see very bad reconstruction error there. And so this is a great approach that for estimating the number of components that's necessary to fit this model, and for estimating the number of factors that are really in the data, the number of sequences, but it doesn't work in all cases. So here's just a few examples of different noise cases and what this cross-validation procedure yields. And so in the top two left, you can see that there's a clear minima at 3 factors for this additive noise and this jitter case. But if you look at this warping case on the right, so this is an intermediate or sort of mild warping noise added, both the generalization error and the training error just go down. And so this one might be evidence that there really aren't 3 components in your warping model. There's really infinitely many because there's all these different speeds of sequence. But even if you look, for instance, at this bottom middle one where you have slightly worse jitter, you can see that although there's some bend at 3 components, there's no minima, and it can be hard to interpret cases like this, which we have seen in real data, where you don't really know whether you have 3 sequences in your data or if there's something else going on.
So basically, sometimes this gives you a clear answer, but if it doesn't, you could use you could choose different lambdas and look at what factors you get in that case. And so, yeah.
Yeah. And so we also tried this on the real data. So you can see here on the left, one of the Songbird HPC datasets. And here it gives a fairly clear answer that there are at least 3 sequences in the data set.
And this is consistent with this is the same data set that I showed a few slides ago, so it's consistent with what we got with the other method. It's consistent with what it looks like by eye, that there's about 3 sequences, and if you try to fit more than 3, then it starts overfitting.
But for the rat hippocampus data set, it was a lot harder to interpret what this means. So maybe there's many more than just a small number? Or
Caution with this technique is that it's a bit nuanced in the sense it depends on how much data you put in, because if you have less data, you might have more overfitting potential. And so I would say if you run it and you get a clear answer, then you can use this, but it really depends on the data set about what type of overfitting you're going to see when. And so I would try both this and try different lambdas like we discussed before.
The other thing that you can do is you can sort of combine the approaches that we talked about. So earlier we showed you how we were using a sweep of the parameter lambda across many orders of magnitude to estimate sort of a best lambda. And we did that using the reconstruction error and this correlation cost term. But you could also imagine using the test error using this masking cross-validation technique. So here in orange is the test error as a function of the parameter lambda, and in blue is the training error as a function of the parameter lambda. So here you can see that as you make lambda smaller, you find a minima at a particular lambda, and then you see that you get massive overfitting as lambda gets very small and you have no regularization or penalty to prevent.
And in many cases, this minima is the same as the lambda that we would choose with our other method. So that's sort of a good validation of our other method in the sense that it's giving you an answer that's similar to what you would do if you were trying to find the best cross-validated performance. And sometimes the cross-validation can get confusing, but yeah.
Mm-hmm. Okay, so yeah.
In terms of just, I call it regular CPU, how long does one cycle of this take for, say, 20 minutes of data with 60 rows of whatever?
20 minutes
But
of data.
It depends on your sampling rate.
Yeah.
And so the data sets that I've been running on, like the movies I showed you of it running, is all slowed down a lot. It runs pretty fast, but like the data sets I've been running it on, which are 100-ish neurons by a couple thousand time bins, will run usually less than a minute. But yeah, it depends also on the number of factors and the L as well.
So the hippocampal data is like, it's about 25 or 30 minutes of data. And that will run with maybe 10 factors, each factor having an L of 500 time bins. That runs in about 30 minutes. But the sampling rate that I was using it's sampled at about 12 hertz. So if you want, much finer sampling than that, then it will take longer than a half hour. But yeah.
So is there any, I guess, tricks? Because in the case of both optimizing lambda and optimizing the number of filters, it's going to be two-dimensional cross-validation optimization. It takes 30 to 40 minutes for the hippocampal data set at 12 hertz. And I think most would probably be 1,000 hertz just because you get millisecond granularity. Is that going to be like weeks of computation? Is this on a CPU or a GPU?
It's on a CPU. It's not a two-dimensional optimization because you're just sweeping lambda and you're just running it at different lambdas.
Lambda is not completely independent of the filter, presumably.
Of-
So you can have different filters and lambdas which give you the better fit.
Sorry, what do you mean by the filter?
When you're trying to optimize the filter size, and then you're also trying
Oh.
to optimize lambda, doing them together, because maybe a lambda of 10 to the 0 is great here, but with the filter 3, maybe it's all or something.
Yeah, we can talk about this a little bit more on the end. Basically, there has been some work making an online version of convolutional NMF, and so if you really needed it to be faster, like algorithmically, you could run it in an online fashion, which is basically on smaller sets and aggregating them. Yeah, so there are sort of ways of making the algorithm faster, but we just haven't done that yet. So
Yeah.
yeah.
It also might not be necessary to sample as finely lambda as I
Yeah,
did.
you can be more. There are strategies. We could just talk about it more. Yeah, yeah.
We also find that once you've chosen one for a particular data type, you can really use that lambda pretty robustly for another data set of the same type. So like when we optimized for calcium imaging data from HVC, we find that we can sort of use the same parameters for all the different data sets that Emily has without having to keep doing this. Anyway.
Yeah, remember, it's usually several orders of magnitude that will be fine. Okay, let's quickly go through parts in event space, because I think this is a cool point, but we also don't want to keep you guys too long. So let's say you have data that looks like this. There's two ways that you could describe this data set. You could say you have two different types of events, this one and this one. Or alternatively, you could say that you have two different sequence parts. So you could say that you have this ensemble and that ensemble. Sometimes only this one happens, and sometimes they have to happen together. So these are two different ways you could describe the data, ways you could factorize the data, and they both give you exactly the right answer in terms of reconstructing the data. It might just be a personal preference of which one you prefer. For example, if you're trying to break your neurons into cell types, you might prefer a part-spaced factorization, whereas if you're trying to correlate neural events with behavior, then you might prefer an event-spaced factorization. So you might want to have some control over which factorization you get in the end. Mm-hmm. And one thing to notice is in the part-spaced factorization, there are correlations in the H's and the time courses, but there are not correlations in the W's and the patterns. And vice versa, in the event-spaced case, there are correlations in the patterns, but not in the time courses. And so making use of this observation, you can just toggle between these by punishing either correlations in the time courses exclusively or exclusively correlations in the neurons. So here's a simulated data set with just like the cartoon that I showed you. And you could do it either way. And if you run convolutional NMF with two factors, then it will just decide to give you the event-spaced one. But let's say you really wanted to correlate it with behavioral events. So you really preferred a parts-spaced factorization.
Event-spaced?
events-based factorization, you could penalize H correlations and get this events-based correlation. And so,
and likewise, here's another data set, but here there's several more different parts-based. So here there are 6 different ensembles of neurons that form a sequence, but there are only 3 combinations on the right. So you can see the events-based has 3, and the parts-based has 6. And again, we can toggle between them. So if you run just regular convolutional NMF with 3 factors on this, you'll get this events-based. But if you penalize the correlations in the W, then you can get a factorization which gives you individual factors for each of these ensembles.
So you can just choose whichever you want. And I tried this on my data that I showed you. And you can get event-based, so you can see these H's are very uncorrelated. This would be great for correlating with the song, see which happens at which syllables. The parts-based, this would be great for clustering the neurons into groups. But these traces are still correlated with each other.
Okay, so the last thing that we wanted to just mention was just sort of an idea about how you might use this for denoising. So,
and it's sort of an idea and a caution, because oftentimes people use PCA for denoising, which works great in some cases, but if you have sequences in your data, it can actually mess up your sequences.
Yeah, so here, I mean, often people will perform dimensionality reduction and throw away some of the smaller components. But if you think that there's repeated temporal structure in your data, you have to be careful because you might end up throwing it out. And so here's some noisy data with 3 sequences on the left. And in the center, you see an SVD reconstructions with different numbers of components. So the top, you see an SVD was performed on this data, and then there's a reconstruction using just the top 3 components of this SVD, 10 or 30 components. And then on the right, you just see convolutional NMF run with either 3 or 10 components. And what you can see is that although you are maybe keeping a lot of the variance when you have 10 components in your SVD model, you're really destroying the temporal structure that was in your data to begin with. Whereas if you use something like convolutional NMF,
is distorted
yeah.
When you just keep the principal components, because you've just kept synchronous activity, and your repeating patterns are not synchronous.
Mm-hmm. And likewise, if you use convolutional NMF to do something like this, then you're more likely to keep things that were repeating of the order of the length of your factorization. And so this can be a way that you can get rid of some of the noise while keeping the structure, whereas with SVD, you would need to use all the components, like the same number of components as your neurons, and you'll keep both the structure but the noise.
Yeah, and this idea of denoising and pattern completion was something Paris Smaragdis was thinking about originally when he was developing convolutional NMF. Basically, sort of, you can read some of his papers about this, but it could do a pretty good job of pattern completion and sort of just keeping the repeating patterns and not keeping a lot of the noise. Okay.
Okay.
So for the exercises, we've posted the code, and we encourage you to go through it, try it on a new data set. Please email us if you have any questions about stuff, or ask us here if you have any questions.
There's
about
a demo
this.
script that you can just download and run all of these different things that we talked about.
And yeah, I guess finally, we are excited to see you guys trying this on a lot of cool data sets. And yeah.
So we're finished. If you'd like, you guys can stick around and try to work through the code, or talk to us about the method or anything else. Otherwise. Hey.
That's really good.
Oh, thanks.
So it seems like